Hi, I'm Dr. Kurt Woeller for Integrative Medicine Academy. The title of this short video is the Organic Acids Test, Quinolinic Acid, NF-kappa B, and Neuroinflammation. If you've done any testing through Great Plains Laboratory or even some of the other labs that carry organic acid profiles, there is commonly a marker on that profile called quinolinic acid. Now, on the Great Plains test, it's actually under the neurotransmitter section. And specifically in this case, it's under the tryptophan metabolites because quinolinic acid is linked to a pathway called quinolinic acid or the chynorinic quinolinic acid pathway. And it's often elevated in people with chronic stress. Now, that chronic stress could come about because of chronic infection. A niacin deficiency sometimes can cause it, and even excess cortisol production through HPA axis engagement. So it's something to watch out for because it is a potent neurotoxin, and then in advanced cases, it can be a contributing factor to different types of neurodegenerative diseases. So this is a slide that's just looking at one aspect of the potential for generating quinolinic acid. So it turns out that tryptophan in our diet, which will typically go off to produce serotonin, but under things that activate these enzymes, the TDO and IDO, the indoleamine dioxygenase enzyme, for example, which can be triggered by various stressors, will pull tryptophan into the quinolinic acid pathway, or at least quinolinic acid gets produced. And one of the reasons is if we have a deficiency in nicotinamide adenine dinucleotide. Now, niacin can help fill the gap as well as niacinamide. And you'll notice that niacin, it's interesting, niacin actually has to pass up into nicotinic acid mononucleotide, which passes through or down through nicotinic dinucleotide and then eventually down into NAD, whereas niacinamide is a little bit more of a straight shot. Now, there's many things that can lead to elevated quinolinic acid, as I mentioned before. Quinolinic acid, interestingly enough, is a metal chelator, and it inhibits various enzymes in the body that regulate or have an effect on glucose. So there's a lot of other things going on with quinolinic acid that are important to know about. This is one of my favorite slides. I use it in many of my lectures, and we talk about this quite extensively in the Advanced Organic Acid Mastery course that we have through Integrative Medicine Academy. So let me just set the stage here real quickly. Here is quinolinic acid. This is an NMDA receptor. So this is an excitatory receptor in the brain. And quinolinic acid has a positive effect. So it'll stimulate that receptor that will cause an influx of calcium into the cell. An influx of calcium, when things are not regulated, can lead to a lot of increased nitrogen-oxygen species that generate oxidative radicals. Now, those can have a damaging effect on mitochondria. When we damage mitochondria, we express cytochrome C. Cytochrome C activates caspase enzymes that lead to apoptotic death of the nerve cell. Increasing calcium influx into the cell because of quinolinic acid stimulation can decrease glutathione, can activate lipases, express arachidonic acid within the cell, and that itself can lead to oxidative reactive species. And these reactive oxygen species get outside the neuron, and they themselves can compromise glial cells, which make up a lot of the structural component in the brain. And one of the things that glial cells do, like astroglia, is they will regulate glutamate in the synaptic space between nerve cells, because glutamate itself is also a positive trigger on NMDA receptor activity. So when we have too much oxidative stress compromising glial cells, that causes a decrease in glutamate regulation. Now, it turns out that quinolinic acid also will inhibit glutamate uptake. Quinolinic acid can bind with iron that's generating hydroxyl radicals that itself causes destruction of the outer membrane of the nerve cell. So as you can see, there's a lot of things happening here with excess quinolinic acid. Now, this was an interesting paper, came out in Biological Psychiatry 2009, called Inflammation and Its Discontents, the Role of Cytokines in the Pathophysiology of Major Depression. So let me set the stage here real quickly on this slide. We've already talked about reactive oxygen species, okay, having an effect on expression of, or not expression of glutamate, but the inability to regulate glutamate through the astroglia cells. So here's an astrocyte. Now, on the other hand, we also have what are called macrophages, these resident, microglia, which are resident macrophages in the nervous system. And when they are triggered, they can stimulate IDO activity, which takes tryptophan away from that pathway and moves it into the production of quinolinic acid. Now, quinolinic acid, we know, can have a negative effect on astrocytes' ability to regulate glutamate. Excess glutamate causes increased positive stimulation of the NMDA receptor. That causes all of those things I mentioned before. It also causes a decrease of something called brain-derived neurotrophic factor, which helps with neuroplasticity. So we're basically getting decreased plasticity, decreased flexibility in the brain. It also compromises neuron formation or new neuron formation and repair. When we compromise astrocytes, we're going to decrease trophic flow. Trophic flow is all about survival, differentiation, and growth within the nervous system. And trophic flow also aids oligodendrocytes in their production of myelin, which is necessary for the myelination of axons, nerve cells in the brain. And so that becomes compromised. The other thing is when we have excess reactive oxygen species by microglia activation, we also get cytokines that get produced. These are different immune chemicals which can have pro-inflammatory effects. Now, cytokines can get expressed in the nervous system or expressed also within the bloodstream by various cells that are circulating through the bloodstream. So up in the bloodstream, we have cytokine passage through the leaky blood-brain barrier, or that can cause leaky blood-brain barrier, that triggers more microglia activation. And whenever we're stimulating pro-inflammatory chemicals in the brain, one of the things that gets triggered is something called NF-kappa B. Now, NF-kappa B is a type of transcription factor or communication messaging system or chemical in the cell. So it basically gets activated by various things at the cell membrane. And this NF-kappa B, as this group of what's called P50, P56 protein combination, comes together and then activates through receptor mechanisms on the DNA that then triggers the production of messenger RNA types. And in this particular case, it's sequencing messenger RNA, which then produces inflammatory proteins. So the way it works is that NF-kappa B activates DNA to make messenger RNA. The messenger RNA moves into the cytoplasm of the cell, complexes with ribosomes, which then produce pro-inflammatory cytokines. Now, cytokines like interferon alpha, interferon gamma can decrease central nervous system concentrations of something called BH4. And so we talk a lot about this in the organic acids test course for advanced concepts, because BH4 is actually a very important cofactor in phenylalanine, tyrosine, and tryptophan hydroxylase. So if we don't have enough BH4, we're going to have a problem in making dopamine as well as serotonin, which also means if we don't have enough BH4, we're going to leave more tryptophan around to be converted into quinolinic acid. Now, there's an enzyme called nitric oxide. It turns out that BH4 is a cofactor for nitric oxide synthase for the production of nitric oxide. Okay, so because we obviously need some nitric oxide, it helps with blood flow, for example. But when we have excess cytokine production because of inflammatory triggering in the brain, we get a decreased concentration of central nervous system concentrations of BH4 and dopamine, for example, which can contribute to depression because of imbalances in nitric oxide stimulation. So as I mentioned, BH4 is an enzyme cofactor for tyrosine hydroxylase, which is important for the development of dopamine. But BH4 is also required for tryptophan hydroxylase, meaning it converts tryptophan into 5-HTP for eventual production of serotonin. And it helps convert phenylalanine hydroxylase into tyrosine for dopamine production. So if we have too much pro-inflammatory activity in the brain that is essentially using up BH4 through the activation of nitric oxide production, we're going to run a deficit of BH4 to help in other hydroxylase enzymes that are involved in dopamine and serotonin production. And so this is one of the contributing factors to depression and one aspect of how neuroinflammation can lead to depression. And so microglia, again, those cells, they're macrophages that took up shop within the brain. They're essentially what are called resident macrophages. They're there to attack and go after things that get into the brain that normally shouldn't get there. And so there's many things that can trigger microglia, like beta amyloid proteins in Alzheimer's, for example, lipopolysaccharide, which comes from gram-negative bacteria. And then these microglia get activated. They then produce, or there is a production of different types of chemicals. So TNF-alpha, for example, hydrogen peroxide, superoxide anions, nitric oxide species, for example. And they themselves will have subsequent damage to other nerve cells in the brain. And then essentially we get locked in what's called reactive microgliosis. So activation of microglia is associated with increased nitric oxide production, which can lead to a lack of available BH4 for other hydroxylase enzymes. So think about that. If we're activating microglia because of infection or things that are passing through the blood-brain barrier that have an immunogenic effect, we're going to increase nitric oxide levels in the brain, but in doing so, that nitric oxide needs BH4 to be produced, but it's going to preferentially take the BH4 reserves, leaving less BH4 available for phenylalanine, tyrosine, and tryptophan hydroxylase to make their respective neurotransmitters, which would be dopamine and serotonin, for example. So this is one aspect of how neuroinflammation can lead to depression. Where another link to this is that microglia also get activated by quinolinic acid. That's something that we can pick up on from the organic acids test. So we have an advanced OAT mastery course through Integrative Medicine Academy. This is taking the organic acid test, primarily Great Plains organic acid test, but when you go through this course, you'll also be able to interpret other companies' organic acid profiles as well that exist within the integrative functional medicine world. And we go through every marker in a very in-depth way and make all different kinds of clinical correlations to what these markers mean. So for more information about the Advanced OAT Mastery course, go to advancedoatmasterycourse.com. As healthcare practitioners, whether you're a health coach, a nutritionist, an acupuncturist, chiropractor, naturopath, DO, MD, you have access to us. That would be myself, Dr. Woeller, my partner, Dr. Trincatella, through our functionalmedicineclinicalrounds.com website. This is where we can consult with you regarding lab tests, case presentations, clinical troubleshooting, give you our insights into things. And there's a way you can actually schedule one-on-one consults with us. We also have some educational information on this membership site as well, including these one-page downloadable clinical data sheets. These are PDF documents that are downloaded one page, that there's a whole archived library of these things. And these are clinical pearls, or these are a lab test interpretation, or clinical information regarding a lab marker. There's all kinds of stuff within the website. So for more information about functional medicine clinical rounds, you can go to the website. And if you have any additional questions about our mastery courses through Integrative Medicine Academy, please go to integrativemedicineacademy.com, or you can email us at integrativemedicineacademy@gmail.com. Again, I am Dr. Kurt Woeller for Integrative Medicine Academy. Thank you.